Hello students, in today's class we are going to learn about cell cycle regulation. So cell cycle is basically the ordered sequence of events that occur in the cell in preparation for cell division. Cell cycle can be broadly categorized or classified into the interphase and the mitotic phase. The mitotic phase is the actual cell division phase, which can be further divided into karyokinesis and cytokinesis. Karyokinesis includes division of the chromatin or the genetic material that is divided into prophase, metaphase, anaphase, and telophase, while cytokinesis represents the division of the cytosol. On the other hand, interphase is basically the phase in between two mitotic stages or the cell division stages, and it is basically a preparatory phase where the cell prepares for the mitotic phase. The interphase can be divided into three main subphases, that is G1 phase, S phase, and G2 phase, growth phase 1, synthetic phase, and the growth phase 2 respectively. Coming to the growth phase 1, this is the longest and the most variable cell cycle phase. So when the cells are born at cell division or cytokinesis, they are about half the size of their normal size, and therefore before the next mitotic division, the cell needs to grow back in size to its optimal original size, and during the G1 phase, basically the cell grows back to its original size. Many activities involved in the cell cycle progression are repressed during the G1 phase so that the cell cannot initiate a new round of proliferation unless it is ready. This repressive control system is known as the R point or the restriction point. The cell can, however, pass through the restriction point in response to certain triggers or signals that indicate or signal the beginning of the new cell cycle division phase. So these triggers are highly regulated. However, if there is misregulation in the regulation process, then it can lead to uncontrolled cell division, which is also known as cancer. Sometimes from the G1 phase, the cell may enter into the G0 phase. So here the cells are basically not dividing, but they enter into this specialized phase, which is known as the G0 phase, where they differentiate to carry out specialized functions and the cell is no longer dividing. Here the cells are not dormant either. They are actually actively involved in protein synthesis and secretion process. This phase is not permanent, and in some specialized cells, the cells from the G0 phase can re-enter into the cell cycle again in response to specific stimuli. This stage is also highly regulated, as again uncontrolled proliferation of cells can lead to cancer. Now coming to the S phase or the synthetic phase, this is the stage where replication of DNA takes place by beginning the process at the origin of replication. Each region of the chromosome that is replicated from the single origin of replication is known as a replicon. Each origin of replication is prepared for the replication by formation of a complex, which is known as pre-replication complex during the G1 phase. This process is also referred to as the licensing stage that ensures that the replication process or the synthesis of daughter DNA will take place only once during each cell cycle. At each origin, fires basically means that begins the replication process during the S phase, and then the pre-replication complex is dismantled and it cannot be reassembled again until the next G1 phase. This basically ensures that the origin will begin only once, the replication will begin at the origin only once during the cell cycle. G2 phase, also known as the growth phase 2, is a brief or a short phase during which key enzymatic activities that will trigger the entry of the cell into mitosis will take place. When these activity reaches a critical threshold level, the cell will enter into the mitosis. Here, the chromatin and cytoskeleton are prepared for dramatic changes that will occur during the mitosis phase. If the DNA is damaged and the damage is detected during the G2 phase, a checkpoint will be activated and the DNA damage response is being triggered that delays the entry of the cell into the mitotic phase. Now coming to the regulatory proteins that overall play an important role in controlling the cell cycle are known as cyclins and CDKs. So cyclins are proteins that undergo a cyclic pattern of synthesis and degradation in a time-bound fashion, and hence they are known as cyclins. CDKs, the full form for it is cyclin-dependent kinases. These are basically kinase molecules that can phosphorylate other proteins. However, these cyclins and the CDKs, they function as one unit, and hence they are often known as Cyclin-CDK complex. So coming to the main functions of cyclin-dependent kinases, these function as serine-threonine kinases, that is, they are going to phosphorylate the serine and threonine residues in the target protein. The activity of these proteins are dependent on cyclin proteins. These CDKs can regulate cell cycle by phosphorylating the target proteins involved in various cell cycle events, and the activity of Cyclin-CDK complexes is regulated by other kinases and phosphatase protein action. Now coming to the different types of Cyclin-CDK complexes. So depending on the four different phases of the cell cycle, there are four different types of Cyclin-CDK complexes that are active. The G1 phase Cyclin-CDK complex is known as Cyclin-D1 CDK4/6 complex. The S phase Cyclin-CDK complex is known as Cyclin-E1 CDK2 complex. Cyclin-E1 CDK2 complex. Cyclin-E1 CDK2 complex is the complex involved in G2 phase, while the cyclin D1 CDK1 is involved in the M phase. Now coming to the first complex, that is cyclin D1 CDK4/6 complex. This is the G1 phase cyclin. It is activated in presence of triggers or signals such as growth factors that results in formation of an active cyclin D CDK4/6 complex. So this complex will take important decision on if the cell can pass through the R point or not. R point is a point in the G1 phase. Once the cell has passed through the restriction point or the R point, then the cell will definitely divide. Whether the cell can pass through the restriction point or not depends on factors such as presence of growth factors, the good nutritional status of the cell, and absence of stress factors. So this decision of whether to pass through the R point or not is dependent on the activity of Cyclin-CDK complex of the G1 phase. However, this protein doesn't function directly, but it requires help from another protein, which is known as the RB1 protein, that functions to regulate the cell cycle progression through the R protein, through the R point. So RB1 protein is basically a tumor suppressor protein. It will suppress the growth. The PRB protein is encoded by the RB1 gene, which on transcription and translation will synthesize the PRB protein. PRB protein basically acts as at the R point. However, it also doesn't directly act on the R point, while what it does is PRB protein can basically bind and inhibit another protein, which is known as E2F protein. E2F protein is important and is required for the synthesis of S phase cyclin. Now, for E2F to regulate the S phase cyclin synthesis, it needs to be dissociated from PRB protein. So here we have the role of the cyclin D1 CDK46 complex that is involved in phosphorylation of PRB protein. On phosphorylation, it undergoes a structural change leading to dissociation from the E2F protein. E2F protein, on dissociation from the PRB phosphorylated PRB protein, it can bind and act as a regulatory protein required for expression of the S phase cyclin. Once the S phase cyclin is synthesized, the cell is already in the S phase or the synthetic phase, during which the replication process will occur. There is another regulatory protein that is also involved, which is known as P53. This is also another tumor suppressor protein. This can modulate the activity of cyclin-CDK complex and hence can regulate cell cycle. The P53 protein plays an important role in cell cycle regulation, apoptosis, and also in maintenance of genome stability. It can affect the cell cycle progression in many ways. Coming to the mode of regulation by P53. So P53 becomes activated when there are some kind of DNA damage that is present on the DNA. And this damage is identified by proteins known as DNA damage sensors, which include the ATR and the ATM proteins. Once these sensors recognize the damage, they bring about activation of other downstream proteins such as the CHEK2 that will further bring about phosphorylation and activation of P53. The tumor suppressor protein P53, on the other hand, can bring about activation of other proteins such as P21 that have an inhibitory activity on cyclin-CDK complex. And in this way, because of the inhibition of activity of cyclin-CDK complex, the cell cycle will take a halt or the cell cycle is paused in the synthetic phase. So here it will not proceed unless the repair is been corrected normally. So so these same points are mentioned here about the role of P53 in the slide. So there are basically the DNA repair that will take place here after the pause of the cell cycle, which can happen by two different ways, either homologous recombination based mechanism or non-homologous end joining based repair mechanism. Now, after the DNA repair procedure, there are two results. Either the DNA damage is being repaired. So if the damage is repaired, then the cell cycle is resumed and then the cell can continue with the stages of cell cycle. However, there are cases where the damage is enormous and it is beyond repair. So in that case, the DNA is not repaired. And here there are two options for the cell. Either the cell can trigger an apoptosis like reaction that will lead to programmed cell death. So in other cases where apoptosis is not triggered by mistake, these fault or mistakes in the chromosome are segregated and this can then later on lead to uncontrolled cell division or a faulty cell division leading to cancer or tumor formation. Next, we are going to discuss about the S phase cyclin, that is cyclin E1 CDK2 complex, which plays an important role during the synthetic phase, and it also ensures that the replication happens only once during the cell cycle. So the replication will begin by binding of a replicator sequence that is named here as ORC. So this replication complex will bind to the origin, and then followed by this there will be also binding of other proteins such as CDT1 and CDC6 proteins. Followed by this, there is also binding of the MCM2 to 7 complex. So the MCM2 7 complex is basically helicase protein that plays an important role in unwinding of the DNA and formation of the replication bubble. So to begin the process, here we have activity of the S-phase cyclin E1 CDK2 complex, which is involved in phosphorylation of CDT1, CDC6, as well as the MCM27 complex. After phosphorylation, they become activated, and you will have unwinding of the DNA, resulting in formation of the replication bubble. And the replication enzyme has already begun the process of replication here. After the beginning of the replication process, there will be dissociation of the ORC as well as the phosphorylated CDC6 and CDT1 protein. So here we can see that the CDT1 and the CDT6 are basically present in a hyperphosphorylated form towards the end of the synthetic phase, and as a result, in a when it is already phosphorylated, it is unable to bind again at the origin and begin the with another round of replication. So this basically ensures that the replication process will happen only once, and eventually these proteins will get dephosphorylated, and only at the towards the end of the next cell cycle, that is in the next G1 phase, only they can be assembled again at the origin. So in this way, the S-phase Cyclin-CDK complexes, they will ensure that the phosphorylation will happen only when the S-phase Cyclin-CDK complex is present of these protein, and hence the they ensure that the replication will initiate only once during the cell cycle. So these are the points, same points that are mentioned here for your reference. Now coming to the last group of cyclin, that is M-phase cyclins, and these also play an important role in cell cycle progression. So in the M-phase cyclin CDK complex is known as cyclin B1 CDK1 complex, and these has various sites such as the threonine 14 and the threonine 16 sites, which act as the inhibitory sites. There are enzymes such as the V1 kinase that bring about phosphorylation of these sites and hence inhibit the activity of the Cyclin-CDK complex, while there are other sites such as the threonine 161 site, which on phosphorylation will activate the Cyclin-CDK complex. These are phosphorylated by the activity of other kinases such as the CAC kinase. Then we also have certain phosphatases such as CDC25 phosphatase, which can remove the phosphorylation that is present on the inhibitory side, that is threonine 14 and 16, which will lead to complete activation of cyclin B1 CDK1 complex. Once the M-phase cyclin CDK complex is active, then it can carry out various functions associated with the cell mitotic phase. This includes, first of all, chromosome condensation. So here for the chromosome condensation, the cyclin B1 CDK1 complex is involved in phosphorylating certain proteins which are known as condensin proteins that will promote chromosome condensation at the beginning of the M phase. Similarly, we also have the activity of cyclin B1 CDK1 complex that at the beginning of the prophase will phosphorylate the lamin proteins, which are part of the intermediate filament that support the nucleus. And because of the phosphorylation event, that leads to breakdown of the nuclear envelope, which is again important process required during nuclear division. Followed by this, they also control other activities such as fragmentation of the cell organelles like Golgi and formation of the spindle apparatus. So the same points are mentioned here about the activity of M phase cyclin-CDK, cyclin B1, CDK1 complex for your reference. So that's it about the role of different cyclin-CDK complexes. Thank you, and kindly like, share, and subscribe if you like the content.